All right, let's talk about treating severe genetic blood disorders in babies. Things like these really tough immune deficiencies or bone marrow failures. It's incredibly challenging, right? These complex procedures, significant risks right at the very start of life.
Yeah, absolutely. These are conditions where, getting in early can make a massive difference. But the current methods, things like gene therapy, well, they can be pretty invasive.
Right, quite invasive. And they involve a lot.
A lot of prep, a lot of resources, yeah.
So the current approach, the successful one, often involves taking cells out, modifying them outside the body, and then putting them back. But what if? What if there was a simpler way, maybe less invasive? A way to get that gene therapy to the crucial blood stem cells, use it in like a natural window of opportunity that just happens right after birth.
That's exactly the exciting possibility that this new research is digging into.
And that's what we're doing our deep dive on today. We're looking at a study from Nature, from researchers at the San Raffaele-Telethon Institute for Gene Therapy. SCID, they're often called in Milan. These guys are really leaders in the field.
Oh, absolutely. SCID is, well, world-renowned for their gene therapy work, especially with lentiviral vectors. They've got a real track record of taking lab discoveries and actually getting them into clinical use.
So our mission today is to guide you through this particular study. We want to understand what they actually found about this early postnatal window for gene therapy. Why does it exist? How did they test it? And, what could it really mean down the line for treating babies with these severe disorders? Let's get into it.
Okay, sounds good. So, just to set the scene a bit, right now, for many of these genetic blood disorders affecting stem cells, the kind of gold standard approach is ex vivo gene therapy.
Ex vivo. And SCID, as you said, they've had real success here. How does that actually work, the ex vivo part?
Right, ex vivo just means outside the body. So essentially you take the patient's own blood stem cells out.
Harvest them.
Yeah, harvest them. Then you process them in a highly specialized lab. Often that involves using those lentiviral vectors we mentioned to insert a working copy of the faulty gene.
Okay.
And then after expanding those corrected cells, and often after giving the patient chemotherapy, which clears space in their bone marrow, you infuse the modified cells back into the patient.
And we know this approach can be really effective. The source mentions SCID developed the gene therapy for MLD, metachromatic leukodystrophy, using this ex vivo method, and that's approved treatment now in Europe and the US.
That's a perfect example, yeah. Shows its power. But, even though it works, it is inherently a complex process.
Yeah.
You've got the cell collection, the really sophisticated lab work, sometimes that conditioning chemo,
Right.
And then the reinfusion. It's invasive, needs very specialized facilities, expert teams. It's a big undertaking.
Which naturally leads to looking for alternatives. And that brings us right to this new study exploring a different path in vivo gene therapy, delivering the therapy directly into the body.
Exactly. And the really fascinating finding in this paper, the core discovery, is that there seems to be this specific time frame very early on, right after birth.
A window.
A window, yeah, where you can actually deliver these gene therapy vectors straight into the bloodstream. And they managed to find and modify the blood stem cells effectively.
Okay, this is the key part. Why? Why does this window exist then? What's special about that early postnatal period?
So Dr. Michela Milani, the lead author, explains this. She points out that right after birth, these blood stem cells, they call them HSPCs, hematopoietic stem and progenitor cells, they're actually migrating.
Migrating? Where are they going?
They're moving house. They develop mainly in the fetal liver late in pregnancy, and now they're transitioning to the bone marrow. That's where they'll live for the rest of the person's life.
Ah, so they're literally in transit.
Exactly. And because they're on the move, they're circulating around in the blood in much higher numbers than you'd normally find later on.
Which makes them easier targets.
Precisely. Significantly more accessible if you're delivering a therapy intravenously just into the bloodstream.
So the hypothesis is skip the whole ex vivo collection and lab work. If you time it right during this window, maybe just a simple IV injection could get the gene therapy into those circulating stem cells.
That's the core idea they tested. They first saw this happening very clearly in newborn mice, like in the first couple of weeks after birth.
Okay, mice. But what about humans?
Crucially, yes. To see if this might hold true for humans, they looked for these circulating HSPCs in human newborns, and they found them present in the blood over the first few months of life. It suggests a similar population of accessible cells exists in human babies, too.
Okay, so they identified the window. They have a biological reason for it in mice, and there are hints it exists in humans. But does it actually work? Can using this window treat disease?
That's the million-dollar question, isn't it?
Yeah.
That was the essential next step. They tested this in vivo approach in mouse models that were specifically designed to mimic human genetic blood disorders.
Which diseases did they look at in the mice?
They used models for SCID, that's a very severe immune deficiency.
Right, I've heard of that.
Autosomal recessive osteopetrosis, which is a serious bone disease, and also Fanconi anemia. That's a really devastating bone marrow failure syndrome. And for the Fanconi anemia model, they actually collaborated with Paulo Rio and Juan Buren's groups in Madrid.
Okay, 3 different conditions modeled. What happened when they tried the in vivo therapy during that early window?
The results were really quite encouraging. In all 3 disease models they tested, giving the gene therapy in vivo right in that early postnatal period led to significant therapeutic benefits.
Like what kind of benefits?
They saw improvements in the actual disease symptoms, and importantly, the treated animals lived longer than the untreated controls.
Wow, so it wasn't just detecting gene transfer, it actually had a functional impact on the disease.
It really did. And the Fanconi anemia results were particularly interesting. They saw that the corrected stem cells didn't just show up, they actually took hold and progressively repopulated the blood system.
They took over.
In a way, yes. They prevented the bone marrow failure that characterizes the disease in that model. And the researchers noted this mirrors something seen in human trials for Fanconi anemia too. The healthy, corrected cells have a natural growth advantage over the defective ones.
That's a really positive sign. So they showed it works in principle. Did they try to improve it, make it better, or maybe stretch out that window a bit?
Yes, they looked into optimizing the process too. They experimented with using drugs that are already approved for clinical use mobilizer drugs, they're called. Things like GCSF and plerixafor.
What do those do?
They're used in the clinic to basically push stem cells out of the bone marrow and into the bloodstream to make them easier to collect for transplants, for example.
Ah, so they tried using those to maybe boost the number of circulating cells.
Exactly. And in older mice, using these mobilizers did seem to increase the number of circulating stem cells and appeared to extend that window of opportunity for the in vivo gene transfer. They also did work on refining the lentiviral vectors themselves, trying to make them more stable, more easily taken up by the target cells. Little tweaks for better performance.
Okay, so let's connect this back to human potential. They saw those circulating stem cells in human newborns. How do the researchers feel about actually translating this into treatments for babies?
Well, Dr. Alessio Cantore, one of the senior scientists, points out that this study provides really solid proof of concept. It shows that in vivo delivery, using these vectors to target blood stem cells, is actually a feasible strategy during this early stage of life for these kinds of disorders.
But how does the efficiency stack up? Is it as good as the current ex vivo methods?
That's an important caveat. Dr. Cantore is quite clear that right now the efficiency they achieved in vivo is still limited compared to the established ex vivo treatments. You don't get quite as high a level of gene correction across all the cells.
Okay, so promising, definitely, but maybe not quite as powerful yet. Could it still be enough to make a difference, though?
He suggests it might be. If this level of efficiency can be replicated in human babies, it could potentially be sufficient to provide a real benefit for certain diseases. He specifically mentions severe immunodeficiencies, like ADA-SCID or Fanconi anemia.
Why those specifically?
Because in those conditions, even correcting a fraction of the stem cells can have a huge positive impact. The corrected cells have that survival or growth advantage we talked about, so they can gradually take over and improve things.
Got it. And Professor Luigi Naldini, he's the director of SR Ticket, a real pioneer in this whole field. He had a really interesting point about these newborn stem cells themselves.
Yes, this is fascinating. Professor Naldini highlights something about the intrinsic nature of these very young stem cells. He explains that normally, when you collect stem cells from adult blood, even after using those mobilizer drugs, those adult stem cells typically need some kind of special activation signal, some stimulus applied in the lab, for the lentiviral vectors to get in and modify them efficiently.
But the newborn cells don't need that.
Apparently not, or at least not to the same extent. He notes that during this early postnatal window, the circulating stem cells aren't just more numerous, they also seem to be inherently more permissive, more open, more receptive to getting the gene transfer without needing those extra activation steps in the lab.
Wow, that's a fundamental difference. It's not just about them being in the right place at the right time, circulating around.
Right.
It's also that they're in the right state, biologically ready to accept the therapy.
Exactly.
Yeah.
It really opens up some intriguing questions about their basic biology right at that specific developmental stage.
Okay, so let's pull this all together. Why does this specific piece of research matter to you, listening now?
Well, fundamentally it matters because this study points towards what could be a really significant shift, maybe even revolutionary, in how we might approach treating severe genetic blood disorders in the future.
You can imagine a scenario that's potentially much less invasive, maybe simpler, perhaps just an intravenous injection shortly after birth, instead of the whole complex process of cell harvesting, lab modification, conditioning chemo.
Exactly. All the things required for many conditions now.
And think about the impact. Treating these truly devastating diseases extremely early, maybe even before symptoms really take hold, that could potentially prevent a lifetime of complications.
It could offer children born with these conditions a dramatically healthier future. It really does offer hope for a different, maybe kinder treatment approach.
It definitely reframes what may be possible for early intervention. Which brings us to a final thought for you to consider, building on that fascinating point from Professor Naldini about the permissiveness of newborn stem cells. This research underscores a clear biological difference. Young stem cells seem uniquely open to gene therapy compared to adult ones without needing that extra push. So the provocative question is, what is it about their fundamental state, their biology, during that specific early window that makes them so receptive?
Yeah, what's the mechanism?
Exactly. And could figuring that out, unlocking the secret of that inherent openness, not only help us improve gene therapy for babies, but could it perhaps, maybe, point towards new ways to make adult stem cells similarly permissive? Could understanding the newborn state unlock new possibilities for gene therapy across all age groups?
That is a really deep biological mystery with potentially huge implications. Definitely something to think about.